Welcome to this podcast on FRET imaging. I'm Charis Antoniades on behalf of the Scientists of Tomorrow of the ESC's Council on Basic Cardiovascular Science. I have with me today Manuela Zaccolo, Professor of Cell Biology in the University of Oxford. Manuela is a leading figure in the field of FRET imaging, and she will share with us some tips and tricks on how to perform a good FRET imaging experiment. Manuela.
Thank you, Charis. So this podcast will be composed of two parts. In the first part, I will give you an introduction on the general principle behind FRET imaging and how this technique can be used to study signaling in intact cells. And in the second part, we will go to the lab, to my lab, and we will film a real FRET experiment. Thank you. So what I would like to do today is to, first of all, give you a very simplified description of what FRET is and a description of how we can use this phenomenon to design and use genetically encoded sensors to measure really biochemical events that happen in intact cells, so in the complexity of the intracellular environment. I will then give you a couple of examples taken from work that we have done in my laboratory to give you a flavor of what type of information you can obtain with this type of approach. And in the last part of my presentation, I want to guide you through a few really basic considerations that one should keep in mind when thinking of developing a new FRET reporter. So what is FRET? FRET stands for fluorescence resonance energy transfer, or Forster's resonance energy transfer from the name of the physicist who described the phenomenon. And FRET is a non-radiative quantum mechanical process by which excitation energy is transferred from a donor fluorophore to an acceptor fluorophore by way of a long-range dipole-dipole interaction. So what does this mean? So we've seen this diagram before. If you have a donor fluorophore, you can excite the donor fluorophore, reach an excited state, and as it relaxes to the ground state, it will emit its own fluorescence. If in close proximity of this donor fluorophore there is another fluorophore that can act as an acceptor, part of the excited state energy of the donor can be transferred by resonance to the acceptor, which will then be excited in turn, and it will emit its own emitted light. And we call this sensitized emission. The efficiency of energy transfer is described by this equation, and as you can see, is very strictly dependent on the distance r between donor and acceptor, and it's inversely proportional to the sixth power of this distance, which means that very small changes in the distance between donor and acceptor translate in very large differences in FRET efficiency. The efficiency of energy transfer also depends on this parameter R0, which is the Forster's radius, which is described by this equation, which indicates that R0 depends on the quantum yield of the donor, which has to do with the ratio between photons emitted to photon absorbed. It depends on the parameter K square, which has to do with the reciprocal alignment of the dipole momentum of the two fluorophores. It depends on N, which is the refractive index, and it depends on J, which is the integral of the spectral overlaps. I will come back to this equation a bit later in my presentation, but for the moment, I'd like to simplify even more this concept in practical terms. So what can we do with FRET? So if you have, for example, two proteins or two domains within a protein, and you can label them with two different fluorophores, in this case, the cyan and the yellow variants of GFP. And if these two domains interact and come close to a distance which is anything between 1 and 10 nanometers, then I can excite the donor fluorophore, CFP in this case, which will emit cyan light. Part of the energy from CFP can be transferred to the nearby acceptor YFP, which will then emit yellow light at the expenses of the emission of the donor CFP. Because the efficiency of this process is very, very tightly dependent on the distance, FRET is a very sensitive way of measuring molecular distance with a resolution, as you can see here. We just heard about the optical limit resolution, which is 200 nanometers, and this allows a resolution which is well below the optical limit due to optical imaging. And so we can exploit this phenomenon to measure protein-protein interactions and conformational change. How do we measure FRET? So if we want to use this to measure biochemical events in the cell, we need a way to measure FRET changes. So FRET can be measured in many different ways, some of which are quite sophisticated and require kind of sophisticated pieces of equipment. But for most of the applications that I'm going to describe today, and for most of the application in live cell imaging, there is a relatively easy way of measuring FRET change, which is to measure sensitized emission. And so in this approach, we only need a rather conventional epifluorescence microscope where we can excite the donor at its appropriate excitation wavelength. And then we can collect the emission from the donor and from the acceptor, and we can ratio these values. We heard this morning about the advantages of ratio-based imaging. And so the value of this ratio will depend on the efficiency of FRET, which in turn will depend on the distance, on the molecular distance between donor and acceptor. So what can we do with this FRET? How can we exploit FRET to generate detectors for intracellular molecular events? As you may know, my laboratory is interested in cyclic nucleotide signaling, so the examples that I will illustrate now are based on sensors to measure cyclic AMP. And to generate a detector for cyclic AMP, as for any other intracellular compound, you need, and to exploit FRET for this, you need to achieve a conformational change on binding of this compound. And so in this case, this structure here illustrates the three-dimensional structure of a cyclic AMP binding domain. This in particular is from the regulatory subunit type 2 of protein kinase A, which is one of the natural intracellular sensors for cyclic AMP. So in the absence of cyclic AMP, this structure looks like this. And when cyclic AMP binds here, there are two major changes. This alpha helix moves a little bit closer to the beta sheet, but in particular, this long helix, which is called C helix, move closer to the cyclic AMP binding pocket here to close the pocket. So this conformational change, although may seem little thing, is actually sufficient to quite significantly change the position of a CFP and YFP that have been fused to the amino terminus and carboxy terminus, respectively, of the cyclic AMP binding domain. And so in this particular case, in the absence of cyclic AMP, the two fluorophores are close enough for FRET. And when cyclic AMP binds, the movement of these helices moves the two fluorophores apart, and now the distance here is too high for FRET to occur. And so we can use this as a very sensitive way of measuring cyclic AMP binding by measuring changes in FRET efficiency. So this is just another representation of a very similar sensor where this is the cyclic AMP binding domain, and here longer linkers have been introduced between the detector part of the reporter and the GFPs, and I will discuss a bit more about linkers later. And so one great advantage of this type of reporters is that they are genetically encoded. So we can introduce the sequence that encodes for these reporters in a plasmid or in a viral vector, and we can transfect or infect our favorite cells, and we can achieve expression of this sensor in the cell, intact cells. And this is an example of a neonatal cardiac myocytes that express one of these cyclic AMP reporters. And this is an image that we collect on excitation of the donor. We collect an image in the cyan channel, and simultaneously an image in the yellow channel. And we then generate a ratio image where pixel by pixel, the software will calculate the numerical value of the ratio between cyan and yellow emission, and it will assign to each pixel a color according to a predefined color scale. And in this particular case, the blue-green means low values for this ratio, so low cyclic AMP, and yellow-red means high value for the ratio, so high cyclic AMP. We can then draw an area of interest on the cell, and we can monitor over time the changes in fluorescence intensity in the two channels. Now, this is the yellow channel, and this is the cyan channel. And of course, at the same time, you can measure over time how the ratio between these two intensity changes. And so now this is the readout of the change in the intracellular concentration of cyclic AMP in real time. And I'm sure you can appreciate that the time resolution that we can achieve here is enormously higher than what can be achieved by conventional ways of measuring cyclic AMP in cell, which involves taking the cell population, breaking up the cells, and then measuring cyclic AMP in the soup using an immuno-based approach. So one great advantage of FRET imaging is the time resolution. And the other great advantage is that we can, of course, average the signal over the entire cell, but we can also observe changes in signal in very highly resolved subcellular compartments. So if I'm going to run this short movie in a moment. So this is illustrates an experiment where this myocyte has been stimulated first with a beta receptor agonist, with isoproterenol here. And then we apply, in the second part of the experiment, an inhibitor of phosphodiesterases. So if you observe how the signal changes within the cell, you will notice that in the first part of the experiment, the signal goes up, so cyclic AMP increases. But the increase is not homogeneous in the cell, but is mainly localized in well-defined compartments. And only when we apply the phosphodiesterase inhibitor, now the signal is more homogeneously distributed within the cell. And this is just a static image of a similar experiment. So by using this approach a number of years ago now, we were able to directly demonstrate that cyclic AMP signals through generation of microdomains. So this, again, is a neonatal cardiac myocyte that is stimulated with norepinephrine. And as you can see, this results in increasing cyclic AMP, which is not homogeneous, but is very clearly localized in specific subcellular microdomains. And on inhibition of phosphodiesterases with IBMX, the boundaries between these microdomains are disrupted, and cyclic AMP can homogeneously spread in the cell, suggesting that the phosphodiesterases may play an important role in regulating the spatial propagation of the cyclic AMP signal. So this idea that cyclic AMP is compartmentalized was proposed many, many years ago, over 35 years ago, based on studies on intact hearts and on isolated cardiac myocytes that show that if in a cardiac myocyte you activate the beta-adrenergic receptor, this generates cyclic AMP, and this leads to increased contractility. But if in the same cell you activate the prostaglandin receptor to an extent that generates a similar amount of cyclic AMP, this has no effect at all on contractility. And this observation of the hormonal specificity of the cyclic AMP response has remained really an open question in the pharmacology of the heart for many, many years. So how come that the cell can produce different functional effects on stimulation of different receptors when the signal is carried by the same intracellular second messenger? And in this paper here in 1979, the author concluded discussion with this statement: we are left with an enigma. Two agonists, iso and prostaglandin, that share several primary characteristics, led to very different results. And so by using this imaging approach, we were able to provide a mechanistic basis for this observation. And I just want very briefly to show you how we did this. So in this experiment, we slightly modified our FRET reporters so that we could target them to different subcellular compartments. I don't have now the time to go into the details of how we did this, but suffice to know that we could target the sensor to the so-called pKa type 1 compartment, which is all the sites within the cell where the pKa type 1 isoform normally sits, or the pKa type 2 compartment, which is the ensemble of all the sites where pKa type 2 isoform normally sits. And so by then using these two differently targeted reporters, we stimulated the prostaglandin receptor, and we found that the cyclic AMP signal generated in this way was detected predominantly by the sensor targeted to the pKa type 1 compartment, whereas when we activated the beta adrenergic receptor, the cyclic AMP signal generated was detected by the sensor targeted to the other compartment. And this is summarized here. So in other words, activation of different G protein kappa receptors was able to generate microdomains of cyclic AMP in different sites within the cell. And we also observed that only activation of the beta receptor led to phosphorylation of targets that are important in regulation of excitation-contraction coupling, whereas activation of the prostaglandin receptor did not affect at all the phosphorylation of these targets, as shown here. So in this way, providing mechanistic basis for the observation that only stimulation of the beta receptor leads to increased contractility, whereas stimulation of the prostaglandin receptor has no effect. So by using this approach, this real-time imaging approach was possible to image directly microdomains of cyclic AMP. A number of other experiments supported the original observation that these microdomains are regulated by phosphodiesterases. But of course, for us, the following question was, how many of these microdomains are there in a cell? And what's the size of these microdomains? And is there more than one microdomain downstream of an individual G protein kappa receptors? And to try and tackle this question, we thought that one possible way was to try and increase the resolution of our imaging approach by targeting the sensors to specific and individual macromolecular complexes that are important in the regulation of cardiac myocyte function. And our initial simple-minded approach was, okay, we take the FRET reporter that I described to you earlier, which is a cyclic AMP binding domain sandwich between YFP and CFP, and we fuse to this microdomain a number of proteins that will carry our sensor where we want the sensor to go. And this was very disappointing because what we found, and this is something that someone that works with targeted reporters need to be aware of. What we found is that fusion of the reporter to different targeting domain very significantly affected the efficiency of FRET. And in some cases, fusion with the targeting domain completely killed the ability of the sensor to respond to increases in cyclic AMP. So here we had to take a detour, which took us quite some time, but we designed a new FRET reporter, which is based on a new design where we free the amino terminus and we fuse the YFP in an external loop in the cyclic AMP binding domain. And we obtain in this way reporters that are reliably targeting where we want them to go. And we are now, we have extensively characterized these reporters, and we are now using them to study local cyclic AMP dynamics downstream of different signals in normal, in disease condition, and we are finding very interesting results and sometimes somewhat surprising results. Okay, so all of this was based on cyclic AMP reporters, but you may not be interested in cyclic AMP at all. And there are many FRET-based reporters to detect many different intracellular signals, or in general to detect many different intracellular compounds. There are reporters that allow to measure PKA and many other kinases activity. But you may want to apply this approach to the particular compound you're interested in, or maybe the specific protein-protein interaction that you're interested in, and there is no reporter for that. So should you embark in designing a new FRET reporter? And if you do embark into this enterprise, what are the basic considerations that you should make? So there are some essential requirements for FRET. The first requirement is that there must be a good spectral overlap between donor and acceptor, and that the quantum yield of the donor must be high. So, as I mentioned, the preferred fluorophores for FRET application in real-time live cell imaging are variants of the green fluorescent protein. And over the years, a number of different spectral variants have been generated. And now there are many of them which cover almost the entire visible spectrum. Not all of them are ideal partners for FRET. And you will need to choose pairs that show the appropriate characteristics. And one of these is spectral overlap, which is illustrated here in this diagram. So this shows the excitation and emission, here in blue, spectra for CFP, and in yellow-green here the excitation and emission spectrum for YFP. So for FRET to work, you need the emission spectrum of the donor, in this case the CFP, to significantly overlap with the excitation spectrum of the acceptor. And this works for CFP, YFP, as indicated here by the overlap region in gray. Because the fluorescent proteins derived from GFP have quite broad absorbance and emission spectra, the spectral overlap comes with a price, and the price is the fact that there is a degree of overlap between the excitation spectrum of the donor and the acceptor, which is highlighted here in red, which means that as you are exciting the donor CFP, you also, to some extent, excite directly the acceptor. And the other contaminant signal is the bleed-through of the donor emission here in the acceptor channel, which is highlighted here by this blue, striped blue area. And as you can see here, for the CFP-YFP couple, this is very significant, and it varies a little bit depending on the specific setup that you're using. But for example, we measure it in our systems, and that can represent up to 40% of the signal that you collect into the YFP channel. So one has to be aware of these contaminant signals because this contaminant signal that you collect in the YFP channel, which is due either to direct excitation or to bleed-through of the donor into the acceptor channel, is signal which is independent of any FRET. So it will contaminate your overall signal. And in particular, if you have low FRET efficiency, you may have problems in detecting your signal from the noise. I also mentioned that the quantum yield for a donor needs to be high. And here are listed a number of GFP variants that have been used as pairs for FRET. So the good thing is that all these couple of fluorophores have been tested already. So if you want to design a FRET reporter, you don't need to go through the testing of all of the different possible combinations. And so far, the most popular pairs are CFP-YFP, which has a decent quantum yield and a very good extinction coefficient, who is a parameter that has to do with the ability of the acceptor to absorb light, and also the cerulean Venus. The other parameter that needs to be satisfied for FRET is that the donor and acceptor dipoles must be aligned relative to each other in space. And there is very little, in fact, that the experimenter can do to improve this parameter, because in most cases, the GFPs will be fused at the amino and carboxy termini of the detector domain. And in this way, the GFP have quite a large degree of freedom of movement around the peptide bonds, which means that the orientation of the dipole is averaged overall. And there is, in fact, very little that one can do to try and improve that, unless the GFP is inserted somehow within another structure that fixes the position of the GFP. So really, the key parameter to keep in mind in the design of an efficient FRET reporter is the distance, the intermolecular distance between donor and acceptor. And as I mentioned earlier, this, the efficiency of energy transfer is inversely proportional to the 6th power of this distance. And it depends on the Forster's distance, which is typical for any donor-acceptor couple. And for any GFP variant, the Forster's distance is about 5 nanometers. So because of the dependency on the inverse 6th power of the distance, the curve that describes FRET efficiency over distance, as you can see here, declines in a very steep manner. And the range of distances which are good to measure FRET is very, very limited. So they extend between 0.5 times the Forster's distance and 1.5 times the Forster's distance, which for GFP pairs, that means anything between 2.5 nanometers and 7.5 nanometers. So any intermolecular distance above 7.5 nanometers abolishes FRET, and any distance below 2.5 nanometers will not add anything to your FRET efficiency. So this clearly, I'm sure you appreciate, imposes a very stringent geometrical restriction to the design of any FRET reporter. And so before embarking in designing a new reporter, there are some very basic considerations that can be made to guide in the design of the sensor. And I have to say that it's very difficult to predict in advance if a FRET reporter will work or not. However, there are some basic considerations that may help reduce the risk that the sensor will not work at all. So if we go back for a moment to the cyclic AMP binding domain here, you see here the C helix, which is the helix that moves away from the cyclic AMP binding pocket in the absence of cyclic AMP. How far this helix can move is not really known, because there is no experimental structure of cyclic AMP binding domains in the absence of cyclic AMP. But one can predict that the space that is sampled by this helix is the space highlighted here by this shaded area. And we can calculate the radius of gyration of this structure, which is the radius of the spherical approximation of the size of this molecule. And for the cyclic AMP binding domain, that's about 1.5 nanometers. Now, if you look at the structure of GFP, which is not drawn to scale, you can see that the GFP, as you know, is a very solid structure, is a beta barrel, which measures about 3 nanometers in diameter and 5 nanometers in height. Here is the fluorophore with the orientation here of the dipole shown by this arrow. And so I told you earlier that the distance for intermolecular distance within which FRET can occur goes from 1 to 10 nanometers maximum. And so one important consideration is to appreciate that an intermolecular distance, which is 3 times the diameter of a GFP, is sufficient to completely abolish FRET. And this is good news in terms of the conformational change that you need to go from FRET to no FRET. But this can be terrible news if your detector domain is a larger domain and if the sites where you can fuse your GFP are far apart. And so the position of the GFP can be the two GFP can be so distant to start with that you will never be able to achieve any FRET. The other consideration has to do with linkers. So I mentioned earlier that linkers can be introduced between the detector domain and the GFPs, and this is often done to increase flexibility, so to allow proper folding of the detector domain and to release or help release any steric hindrance that the GFPs can impose on the detector domain. And here again, it's very difficult to predict the final conformation of any peptide when it is incorporated in a more complex three-dimensional structure. But there are some basic rules that one can consider. So, for example, in a non-structured polypeptide chain, the distance between two alpha carbons is about 0.38 nanometers. And if the sequence, the amino acid sequence of your peptide will lead to an alpha helix fold, then one should consider that the pitch between two consecutive turns is about 0.54 nanometers. And if you have an alpha helix, you will have a very rigid rod-like spacer, whereas if you have an unstructured linker, you will have a much higher degree of freedom of movement. And again, the choice between one or the other would depend on the specific sensor that you are trying to build. But so in deciding what is the length of the linker that you want to design and what is the sequence, the amino acid sequence of this linker, as a minimum, you should consider the size of the building blocks of your sensor. So what is the size of your detector? What is the size of the GFP? How far away you want to place your GFP with respect to the detector? To summarize, in designing a new reporter, there are basically two key aspects that one must consider. One is the detector part of the sensor needs to undergo a significant conformational change on binding of the molecule that you want to detect, or a significant conformational change of when this domain forms or comes in contact with another protein domain if you're measuring protein-protein interaction. And the other consideration has to do with the length of the linker and with the position where the fluorophores can be inserted. So here you can see from this schematic that the effective radius, which can be calculated as the sum of the length of the linker, the radius of gyration of the GFP, and half the distance for FRET, which is 5 nanometers. So this effective radius needs to significantly lead to a significant overlap between the donor and the effector fluorophore in condition of high FRET. And the two needs to separate enough not to have any overlap in the absence of FRET. So there is some room to play here with, so in deciding the length of the linker, and there are ways one can estimate what's the overall end-to-end dimension of a peptide linker. And the other very important parameter, which is the position where the GFP can be fused, very often is not something that the experimenter can really change, because in most cases, the only viable sites are the amino terminus and the carboxy terminus of your detector domain, because any other site where you're trying to insert your GFPs may affect the ability of the detector to fold, or it may affect the ability of the detector to function as a detector. So in most cases, the choice of the distance is very limited. But if one makes these considerations, these very simple geometrical considerations on distance, and has in mind a specific domain that is intended to be used as the detector, then one can, with some good approximation, get an idea if the sensor has any chance to work or not. So to conclude, I would like to acknowledge some of the people that contributed some of the experiments that I showed here. Marco and Giulietta, previous postdocs in my laboratories, and Andreas and Nicoletta, which are part of my group now. And Sergio Pantano, who's a collaborator who is an expert in molecular dynamic simulation, who produced the two slides that I showed at the end. He has written a very interesting chapter that will be published soon in a volume of methods in molecular biology, where he discusses practical approaches to develop new threat reporters.